Turn my phone off here. All right, welcome to today's episode or edition of the South Carolina Sea Grant Consortium's Educator Helpline. My name is Kristen Daring, and I am a marine educator here at the South Carolina Sea Grant Consortium. And today I am downtown in our offices to talk to you about some activities that you can do from the comfort of your own home. This is a helpline, so in case you haven't been to any of these previously, I think we're in our 6th week now, which is crazy to think about. But in case you haven't been able to tune into any of the previous videos, I'm going to tell you a little bit about how this is going to work. Over in the comments, we have Evie Bell, one of our educators, who is answering any questions that you may have and posting associated resources that we will be covering. All of the activities and resources that we are going to be looking at today deal with Ocean Literacy Principle number 4. Now, on the wall behind me, I'm going to go walk over to it real quick because, oh, there it goes, it focused. So on the wall behind me, we have Ocean Literacy Principle number 4 written out. This is part of the Ocean Literacy Framework, which is a set of standards developed to help educators guide their curriculum and guide their instruction for teaching ocean literacy. The 4th principle of 7 is the ocean made the earth habitable. Now, everybody watching this is somebody who lives on Earth, so we depend on the ocean to make it possible for us to live. Now, as we're going, feel free to jump in and out. If you need to go do something else, feel free to do that. If you have any questions along the way, please enter them in the comments on the side. We'd love to hear from you. We'd love to hear where you're visiting from, how you found our video, and just anything you have to share about ocean literacy principles and science education. We're going to try to hit some lab activities so we can actually look at some of the chemical processes, the biochemical processes that the ocean has a part in making the earth habitable. We're going to look at an engineering challenge and some art as well, with maybe a little bit of math mixed in there. So try to hit all of those parts of STEM or STEAM. And like I said, they are all going to be centered around Ocean Literacy Principle number 4. On the side there, you can see the link to the Ocean Literacy Framework. So if you need to check that out, if you want to learn more about the Ocean Literacy Framework, feel free to visit that link and get some ideas for how to structure your ocean science curriculum. All right. So like I said, all of us watching this, all of us participating in this live here on Earth, so we need the oceans in order to live. But not only does the ocean continue to make Earth habitable today, it also was the home to some of the first life on Earth. The earliest fossils that we've been able to find were found down in hydrothermal vents in those really warm, deep areas of the ocean, where you have the energy from the earth coming up, that geochemical energy, and then you also have a lot of nutrients and raw ingredients, as you could look at it, to actually come together to create life on Earth. So some of the first life on Earth, those single-celled microbes and organisms, they started out in the ocean. The ocean is a little bit less of a tempest environment, a tempestuous environment than early life on Earth. Earth was kind of a volatile place in its early history. So 3.5, 4 billion years ago, depending on who you ask and how you age things, life did develop down in those hydrothermal vents where they were a little more sheltered from the craziness that was life on the surface of Earth's early history. As life developed and got more complex, we started to see things come into play like photosynthesis, like oxygen. So photosynthesis is something that we depend on as air breathers. If you have taken some deep breaths as you've been doing your online curriculum, online schooling, you can thank some plankton for that. The earliest oxygen in Earth's environment came from these guys right here. So these are cyanobacteria. There's a type of algae that live in the ocean, and they are some of the first organisms to photosynthesize and create that oxygen to populate the Earth's atmosphere with the air that land animals need to breathe, or animals that don't breathe with gills or through any kind of passive transport of oxygen across through the water. So those cyanobacteria were crucial in putting oxygen into the environment, into the atmosphere on Earth, and making it possible for life to move out of the oceans. All of the oxygen that we need as land animals, about half of that is made up of oxygen that is produced by marine organisms. So we really, really depend on the ocean to continue making Earth habitable. When we look for life on other planets, a lot of times what we look for is water, because the way that we know life is that it's based around water, and the ocean for us is how we have come to have life on Earth, and that water is making it possible for Earth to be lived on by people and any other kind of living creatures. The plankton that a lot of people are more familiar with, or especially a lot of kids are more familiar with, may look a little bit like this guy right here. This is a copepod, and that is one that the plankton from SpongeBob was based off of. So if you are trying to get kids interested in plankton, the plankton from SpongeBob is based on a real life plankton, and you can get them a little more interested in tiny organisms in the ocean just by talking about SpongeBob and how it's a real life thing. All right, so in a little bit, we're going to get even more in depth with plankton and what is a plankton and why do they matter so much. But we're going to focus first on photosynthesis. So I'm going to step behind the table here with all of my activities and show you our first one, which is going to be an activity to actually visualize the process of oxygen production through photosynthesis. All right. All of these activities are ones that you can do with materials from your own home. So for this first one, what you'll need are some cups, some clear cups. I'm using Tupperware just because they're nice and evenly sized and I can transport them to the office. You're going to need some water, some dish soap or some type of mild detergent, and some baking soda like this right here. The other thing you're going to need are spinach leaves, or some kind of thick, flat leaf that you can punch holes out of. I don't have any kind of creative caterpillars by my house. I used a hole punch to punch little discs out of that spinach leaf. So your first step is to punch the holes out of your spinach leaf. You want to have little discs. And the instructions for this activity are going to be linked in the side on the comments as well. So first, you're going to punch those holes out of your spinach leaf. Now, spinach or leaves are where plants do their photosynthesizing. The leaf has that nice wide surface area to gather the light from the sun in order to photosynthesize and bring that energy in to make oxygen and sugars that they need to survive. So we're going to use that part of the plant, that leaf. But if you don't have spinach, that's okay, as long as it's a nice, flat, thick leaf. So you're going to punch 10 evenly sized holes. If you have a 3-hole punch like this, that works fine. If you have a single hole puncher, that works fine too. So you're going to have 10 holes for your control treatment and 10 holes punched for your experimental treatment. In here, we have water, about 250 milliliters of water in each one of these containers. And then to one of them, this one right here with the bubbles, I added some detergent. And to this one, this is just plain water. And this one here has our baking soda in it. I think that's backwards. You should probably label your containers so that that doesn't happen. But your first step, punch out these spinach discs. You're also going to need a syringe. We used one of these for a previous activity in our second week of the Educator Helpline. If you didn't watch that, go back and watch our previous videos. But you can get a syringe like this just from CVS or any kind of drugstore around you. So you're going to open up your syringe and put your 10 little leaf discs in here. I already put my spinach in here earlier today, and so I have my spinach in there, and then you want to close up your syringe. This step, we're going to be taking all of the air out of the spinach leaves. We don't want them to have any oxygen right now. We want them to make it on their own. So after you have your spinach discs inside of your syringe, you're going to open up either your plain water or your baking soda water and your detergent water. That detergent is going to help break the surface tension of the water so that your spinach leaves don't float. You're going to get just a little bit of your detergent and then a little bit of your treatment or your control water in there. And I'm going to come up close here for a second and not get water everywhere. But the next step is going to be creating a vacuum inside of this syringe to get rid of all of the oxygen that is inside those tiny little leaf discs. And the way you're going to do that is to put your thumb over the top of it and pull. Now, if you look close, you can see all these little bubbles that are coming out. That vacuum is pulling the air out of your syringe. You're going to repeat that over and over again as it pulls the air out of those spinach leaves. And your goal is to no longer have the spinach discs float. You want them to get all of that oxygen out, and they will start to sink down and not float at the top. This might take a little bit. You just have to keep going until your spinach discs no longer float up to the surface. And as you do it, you can kind of shake. You can see that my spinach discs are starting to sink down a little bit. I'm getting all of that oxygen out of there with that partial vacuum and squirting water on my computer just a little bit. Whoops. All right, so once you have all of your spinach discs float to the bottom and just be suspended without floating towards the top, then you can move on to your next step. So your next step is going to be to put these spinach discs into your cup of water with either your treatment, the baking soda, or just the plain water. As you put them in there, they should sink to the bottom. I put some spinach discs in this one earlier, and they are still sinking to the bottom. And the next step is to let them photosynthesize. You need some sort of light source. You can bring your discs outside if you have just want to use the sun, or if you have an indoor light, an artificial light, you can use that as well. But they need a light source, they need carbon dioxide, and they need water in order to perform photosynthesis. So now that you have your spinach discs in the water, they don't have any oxygen inside of them. They need to have those ingredients. So you gave them light, they have water, they're inside of water, and then they need carbon dioxide. What students are going to do next is to time how long it takes for those discs to photosynthesize and produce enough oxygen to float up to the surface. This can take a little bit of time. It's going to take 10-15 minutes or so, and you want to have them timing it and watching the whole time to see when those discs start floating up and record that time. What they will find is that the container that has the baking soda in it, the disc will begin to float. But in the container without the baking soda in it, they can't float. And the reason for that is because this baking soda has the source of carbon dioxide in it that they need in order to do the process of photosynthesis and to produce that oxygen that will buoy them up to the surface. And they can only do that in the baking soda treatment. No matter how long they wait in that water, they are not going to float to the top. So you can actually visualize that oxygen production in our algae, in our spinach discs as they float up to the surface. You can make this into a graphing activity and mark the time of when those discs come up. And as you repeat this experiment, you can get some nice data about what the rate of production of oxygen is in your spinach leaves. That oxygen that they make is crucial for our life, like I said before. Plankton and marine organisms provide about half of the oxygen that we need on land to breathe. But they have to do that in a relatively small amount of space. Over here, we have a beautiful diagram of our ocean, and only the top 200 meters or so is where photosynthesis can occur. It's called the photic zone. So the light from the sun can penetrate that first 200 meters of our water column, but it can't go past that. But water absorbs and blocks out the light, keeping it from getting down to our depths of the ocean and to our anglerfish down here who has to make its own light. You can tell that I have a future career in illustration and potentially cartography as well. It's pretty impressive, if I do say so myself. So this photic zone right here, that top 200 meters of the ocean, is where about 90% of ocean life is found. It's important for organisms that can photosynthesize to be able to get that light. Without the light, without the carbon dioxide, they cannot perform photosynthesis. So being up at the top of the water column is a really crucial part of being a plankton. You need to be able to have access to that sunlight. To do this, phytoplankton, the plant-like plankton that can do their own photosynthesis, have developed a lot of different ways to stay up towards the surface. This picture here shows a wide variety of different types of plankton. We have all kinds of phytoplankton. You'll notice that most of them are green. That is because they are using that chlorophyll, that green pigment, in order to hold the sunlight, just like plants on Earth. But they have a wide variety of shapes. You might see some long branching colonial organisms. You might see single-celled round organisms, but all of them have these creative adaptations in order to be able to stay up at the surface. So the next activity that we're going to look at is going to talk about how they do that. How can you design a plankton that can stay up at the surface of the water? Now, this is where our engineering challenge comes in. If you tuned into our second Educator Helpline, we talked all about density and we did the clay ball challenge. This is kind of a similar activity where you're going to take something and try to make it stay up towards the surface. To do this activity, all you're going to need is a container of water that's relatively tall and some silly putty, or you can use clay or other things like that that will not fall apart in water. So probably not Play-Doh, but any other kind of water-safe clay works great. And what you're going to have students do is they are going to take their silly putty and make it into a design that they think is going to sink the slowest. So this is a race to be the slowest. This is a race to stay towards the surface. You still want it to float, because as plankton stay at the surface, they can actually get too much sunlight. Too much of a good thing becomes a bad thing. So not only do they need that sunlight, they also need to have access to the nutrients lower down in the water column. So during a day, a plankton might spend part of its time in the top of the water column and part of its day down in the lower areas of the water column, getting all of those nutrients that it needs to perform all of its other physical and chemical processes that it needs. So they need to take this silly putty and design a plankton that they think will take a long time to sink down. You don't want to give them a lot of opportunities to test this before they put it in the water. They need to think about it, design their plankton. They can base it off of real life plankton, so you can show them pictures of different plankton and they can try to base their design on that. And then once they've come up with a creative design for their plankton, then they can put it into the water and time how long it takes to get down to the bottom. So I'm going to put mine in my water. Oop, mine's float. Oh, there it goes. All right, so that wasn't a great design. It floated for a second and then kind of plummeted down towards the bottom. So I probably would not have won that challenge, but you can have them give it a couple of tries and come up with the best design, the most optimal design in order to stay floating or towards the surface as much as possible. That migration that they go through every day, it's not up to the plankton so much. Plankton means wanderer. A wanderer is going around, just kind of being pushed around by the water. Plankton their motion is determined by the movement of the water, the currents and the tides and the flow of the ocean. Some of the plankton that we think of are really, really small, but plankton can actually be pretty big. Depends on who you ask, but some people would argue that the largest plankton is a mola mola, the sunfish that's the largest bony fish in the world. Some people think that that's not quite true, but they are pretty poor swimmers, and they can't really decide where they want to go too well on their own. And so they could be considered plankton. Some larger plankton that are definitely plankton would be things like jellies. Jellies can't control where they move. But typically when we think about plankton, we think about all these little guys. The phytoplankton that make their own food from the sun, and then the zooplankton or animal-like plankton that can't make their own food. All right, so we did our photosynthesis activity. We did our engineering challenge, our design of plankton, in order to try to mimic that diel or that daily migration through the water column up and down. And then our next activity is going to be a little bit more artistic. As you can tell, as I showed you before, I'm a great artist. So if you're intimidated by this, it's gonna be tricky. And I'm totally kidding in that, because I'm not a great artist, and it's still possible to make cool things without being really, really good at art. So the last thing that we're going to do, because phytoplankton and plankton, I think they are some of the most beautiful things in the ocean and potentially in the world. These are diatoms. Diatoms are a type of phytoplankton that make a shell or a frustule out of silica. They're basically like tiny little glass beads that float through the ocean and are alive. And when I was looking at these, it looked to me like things that we made with spirographs when we were kids. I don't know if you guys had these when you were a kid, but this kit right here, this spirograph kit, I went out and bought one for this because I think they're really, really cool, and they look just like diatoms to me. So you got these spirographs, and with the spirograph you can make some pretty cool designs like this, which looks pretty similar to some of those diatoms that you can see in the picture here. So you can use the spirograph to create a diatom, or if you don't have access to a spirograph, and you don't feel like going out and buying them and playing around with it because you're an adult, you can make your own spirograph with things at your own home. It might be a little trickier to make some of the more complex designs, but it's very, very easy to do, and you can still make some cool things. What you'll need for this is a lid from something like I think this one I got from a breadcrumb container. You can use a lid from a Coke or any kind of bottled soda or drink or whatever. Play around with different sizes and different types of shapes in your internal part. Then you're going to need a circular cake pan. And that's about it. And a rubber band. Excuse me, I spoke too soon. A rubber band, some sort of circular lid, or interestingly shaped lid if you want to get crazy with it, and your cake pan. So what you're going to do is take your lid and then put the rubber band along the outside. This is going to provide traction so that it can spin around inside of your cake pan. In my lid here, I poked some holes. I used a nail to do this, but if you have that single hole punch out still, you can use that to make holes in your lid. If you don't have lids lying around, you can use cardboard and make this out of some stiff cardboard as well. All right, so once you have your holes poked in your lid and your rubber band around the outside, you're going to put a piece of paper down into your cake pan. I already did it a little bit here to not to spoil the movie magic of this activity, but then you're going to put your lid down inside with the side of your lid touching the side of that cake pan. Next, you're going to use a marker or a pen or a pencil, any kind of writing implement, and put it into one of those holes. So you're going to put that through the hole and spin it around and just move it in a circle. That's going to make some nice symmetrical lines inside of there and start to create those really cool patterns that we can see in our spirograph or in our diatoms. From there, you can really take it anywhere you want to go. There's such a wide variety of types of plankton in the world that you can pretty much do anything and then find a plankton that looks like that thing. Another fun activity to identify some plankton and maybe find some things that look like plankton around your house. I was playing with my dog and I found this ball right here, which to me looks just like this. It's hard to tell the difference. They're like twins, right? So this right here is called Volvox. It's actually a freshwater algae, but there are saltwater algae that look pretty similar. But this one here I learned about in high school and it stuck with me because it's just so cool looking. So plankton come in all flavors, freshwater and saltwater, and you can have a scavenger hunt where you walk around your house and try to find things that look like plankton, and then use a plankton identification guide to figure out what plankton it is. So you can make it as creative or as simple as you want. You can start out with looking at those plankton. There are tons of guides online to different species of plankton, and then just walk around your house and find some plankton or things that look like plankton to you. All right, the last thing that I wanted to tell you guys about is an opportunity to get involved with citizen science with plankton. Like I said earlier, too much of a bad thing can become, or too much of a good thing can start to be a bad thing. So although we depend on phytoplankton and on these photosynthetic organisms to produce the oxygen that we need to breathe and to survive, too much of them can start to cause problems. We can have things like that are called harmful algal blooms. Basically, you just have a huge amount of plankton that are congregating. It can be because there's more nutrients than there should be in the water. It can be because of a lot of different reasons. But it's important for us to monitor those and to keep an eye on the phytoplankton in our waterways. So if you would like to help out with that monitoring, you can become part of the Phytoplankton Monitoring Network. In the side comments, Evie's going to link to the Phytoplankton Monitoring Network website, so you can go there. All you're going to need is access to a microscope. If you don't have access to a microscope, they might be able to help you find a microscope that you can use around you, but any other materials that you would need will be provided by NOAA, by the Phytoplankton Monitoring Network. So the Phytoplankton Monitoring Network, basically what you do is at least once a week or once every 2 weeks, you take a water sample and examine it under a microscope. I don't expect anyone to be super familiar with different types of plankton. I know that if I'm looking at a water sample, I'm not going to be able to identify all those plankton. And so what the Phytoplankton Monitoring Network staff do is they train you on identification and on how to categorize if something is present, absent, or abundant. So you don't have to be a plankton expert, though you will probably become one. You can sign up for the Phytoplankton Monitoring Network as an individual. You can do it with your school, with your students, and give them a sense of ownership for looking at the environmental quality of the waterways that they live by. So check out the Phytoplankton Monitoring Network to learn how to do phytoplankton science in your classroom or in your home, and you can help participate in citizen science and making our monitoring networks and our management strategies more effective. All right, so I think that is all I have for you guys today in terms of activities. So I wanted to give everyone a chance to ask questions. I have a couple here. So first one is, what is the difference between zooplankton and phytoplankton? I kind of jumped into talking about phytoplankton before I explained that. But again, zooplankton are plankton that are animal-like, things that can't make their own food. You can think about zooplankton like a zoo. A zoo is full of animals. So zooplankton are animal-like plankton. And then phytoplankton are like plants. Phyto means plant, and then plankton, of course, as we said before, means wanderer. So plant wanderers or animal wanderers. So those phytoplankton can produce their energy, produce their food from the sun, whereas the zooplankton cannot, and then they have to eat something else in order to survive. All right, the next question is a copepod, is a copepod zooplankton or phytoplankton? A copepod is a type of zooplankton, so they cannot make their own food. They have to consume phytoplankton. So plankton are typically really small, like we talked about, and they form the basis for most food webs, for all food webs in the ocean, start with tiny little plankton. So without plankton, we couldn't have all of the other amazing organisms in the ocean. So think about your favorite ocean animal, if it's a sea turtle or a dolphin, or maybe you really like copepods, I don't know. But all of those wouldn't be able to exist without our tiny little plankton friends. All right, another question, why are HABs or harmful algal blooms bad? That's a great question. So in the name, it says harmful. So obviously they're bad enough that they are named harmful algal blooms. But things that they can do is they can affect fish populations. When an algae takes up all of these nutrients, it seems like it would be really, really great. It would be producing all kinds of oxygen, but what they can actually do is form a layer over the top of the water and form these anoxic zones, areas without oxygen inside of them. So you can have huge fish kills. Sometimes you'll see giant amounts of dead fish and dead ocean animals washing up on shore. They can also be harmful in terms of what they can do to us. So not only can they have effects on the oxygen levels in the water, they can also, some types of phytoplankton and plankton can produce chemicals that are actually toxic for us. If you think about things like a red tide, if you've gone on vacation and there was a warning on the beach that there's a red tide here, you shouldn't swim in there, because that red tide, those tiny organisms are called dinoflagellates, which is a really fun word. Those organisms are producing toxic chemicals that can cause health problems for people. If you breathe them in, they can get into your system and really cause problems inside of you. So harmful algal blooms are bad for ocean organisms. They're bad for people, and they can have pretty wide-reaching impacts on coastal organisms. So it's important that we keep an eye on them and make sure that we monitor those phytoplankton and don't let them get out of control. Or if they are starting to get too abundant, then we can manage it and we can try to make things better. All right. If we don't have any more questions, it looks like that is all of them. And man, we are at 29 minutes and 53 seconds. That is almost perfectly half an hour. So I'm going to go ahead and sign off if there aren't any more questions. But I want to thank everyone for joining us and for tuning into this week's Educator Helpline. We are going to be back here next week on Thursday at 4 o'clock, same time, same place, different ocean literacy principle. So our next one's going to be really fun. It's ocean literacy principle number 5 is all about biodiversity and life in the ocean. The ocean is home to the largest animal ever known to exist on Earth. See if you know which one it is. We live right now with the largest animal ever known to live on Earth, which is just crazy to think about. And the tiniest organisms like those microbes and those first organisms that developed in the ocean, we have this huge range of sizes of living things in the ocean. So check it out next week. It's going to be all about that biodiversity and the diverse habitats and organisms that live in the ocean. So getting into the really fun, charismatic megafauna, those really cute big animals that we can look at. All right, thanks again. If you have any questions or if you want to check out any of these activities, or if you want to go back and look at our past videos, all of those are on our website. So check them out, check out the resources on our Sea Grant education page. That's scseagrant.org/education. And Evie will post that in the comments as well. So check out all of the resources that we have for you there and join us next week. Thanks everyone. Have a great day.